Hello and welcome to this webinar about AMD assessment using cellular level retinal imaging. Today we have two wonderful panelists with us. So Professor Michel Paques, he is professor of ophthalmology at the 1520 National Eye Hospital in Paris. And Professor Ethan Rossi is associate professor of ophthalmology at University of Pittsburgh Vision Institute. Hello to Michel and Ethan. So we are very excited because they are both leading experts in ultra-high resolution retinal imaging using adaptive optics technology. Michel and Ethan, they pioneered the development of clinical applications of AO imaging in a wide range of disease and especially in AMD. They have led multiple research studies at 1520 Hospital in Paris and in Pittsburgh, and also in collaboration together. And they already published multiple results. So for today's events, they very kindly agreed to share about how they have been using Imagini's adaptive optics camera, the RTX-1 device, in studies in AMD. So very briefly, from healthy retinas, from healthy subjects. So with the RTX-1, you can observe down to the level of single cone cells, and you can also quantify those cells. And with the TFI module, which is a new add-on that we recently added to the product, it is also possible to have cellular views of the RPE layer. So Michel and Ethan, right now, there is a new context around AMD since new treatments for dry AMD are now under development and there are several clinical trials ongoing. So in this context, what would you say is the main interest of AO retinal imaging, such as it's possible with RTX-1? First, I will ask Ethan.
That's a great question. And from my perspective, I think that really the place where high resolution retinal imaging can help us is in having more fine scale monitoring of smaller changes that can be, sort of invisible to the conventional imaging systems that we use with either spectral domain OCT or other types of imaging we do in the clinic. And, this ability to look at changes over smaller spatial scales, we hope, will also allow us to see change over shorter temporal intervals. So greater change over time to detect for these clinical trials that can be very lengthy, because as you know, AMD is a very slowly progressing disease. And so if we had a tool that was able to see these slow changes more precisely over shorter intervals, we could potentially shorten these clinical trials and just, speed the treatments to the clinic faster. So
yes, this sounds really exciting. And Michel, according to you, what would you say is the main interest of AO retinal imaging in AMD?
I fully agree with Ethan. Of course, I will add other specific features. More precisely, in the case of dry AMD, you have the example of patients having foveal sparing. I mean, an island of RPE cells preserving central vision, which is, of course, of utmost importance. And these cells will disappear over time, and ultimately the atrophy will also progress onto the fovea. And I think at the degree of precision necessary, adaptive optics is the only mean for which and with which we can follow up these patients, and as Ethan said, evaluate drugs. And we can even imagine that we can adapt the dose to the observed response with a matter of weeks instead of semesters or even years in some cases.
So this is really exciting perspectives. So let's start maybe digging a little more into the kind of findings you already obtained with RTX-1. So let's start maybe with early AMD. What did you observe using the RTX-1 camera in those early cases?
So